Alright guys, welcome back to Underground Science, and in this video we'll be talking about a second messenger G protein coupled receptor and the protein kinase A pathway. Alright, so let's go and get started. So this is a intracellular mechanism, alright, so especially for any peptide hormones. Alright, so let's go and write that down. So if you see, in this video we're going to focus on the fact that right? We won't go into the details of what specific peptide hormones. We'll just generalize it as a specific or any type of ligand, all right? But we can say that this is generally going to be for any peptide hormones, all right? So any hormones that are made out of proteins, all right? It's mostly proteins. So let's go and get started. So pretty much the role of a second messenger G protein coupled receptor is that a peptide hormone, like we said, can't get through the phospholipid bilayer of the cell. All right, so let's say this is a cell. All right, so let's say this is a cell. And so actually, I have a zoomed in version here, is that you see here that we zoomed in on just this particular like turn of the cell, and we can see a phospholipid bilayer, right? So it's called a phospholipid bilayer is because it's a bilayer, right? We have two layers, one you can see running down here and here on the outside of phospholipids. So it's called a phospholipid bilayer, what most of the cell membrane is made out of, all right? And the phospholipid bilayer contains our hydrophilic heads, all right? So our hydrophilic heads, and our hydrophobic tails, all right? So these are our hydrophobic tails. And by the name, you can tell that the heads, the hydrophilic heads, right, are water-loving heads, all right, because that's what hydrophilic means, water-loving, all right, so they attract to water. However, the tails, these are fatty acid tails, and we know fat doesn't dissolve with water, all right, so these are hydrophobic tails. They don't like water, all right, so that's why they don't mix with water that well. So they don't attract to water that well, unlike the hydrophilic heads that do attract with water. All right, so that's the basic component of the cell membrane. But let's see how this G protein coupled receptor, right, the second messenger intracellular mechanism, how this plays a role with, let's say, peptide hormones. So remember, peptide hormones cannot get in through this phospholipid bilayer of the cell, right, because it's a peptide hormone, right? It's a protein hormone. So it needs some type of receptor on the outside to activate some proteins on the inside of the cell so those proteins can go ahead and do whatever function the outside first messenger ligand can do. All right, so pretty much here's how it works. So we have a G protein coupled receptor here. All right, so we have a receptor right here, just ready to receive a molecule, right? A ligand, a first messenger. So it's called a cell here. Let me go and draw this out first, then we'll talk about it. So let's go and draw a G protein coupled receptor. All right, and then let's go ahead and synapse this, right? It's not an actual synapse, but I'm just saying it just because it kind of looks like one. Okay, so this is our G protein coupled receptor, and so what happens is, let's say we have a peptide hormone here, and we'll call it a ligand, all right? Something that's binding to a receptor. So let's go and draw this as a circle. All right, so this is called a first messenger that binds on the outside, on the G protein coupled receptor. Remember that right here, all right, all this space is the outside of the cell. All right, so outside of the cell, and then all over here is going to be the inside of the cell. All right, so it's binding to the, oops, sorry about that. It's binding to the outside of the cell. All right, so this ligand is. And because it's on the outside of the cell, we're going to call this ligand actually the first messenger. All right, because it's kind of like the first messenger. It's the first thing that's trying to communicate with the cell. So it's going to be the first messenger because it's outside of the cell. And I know the title of this video is second messenger, right? And if we stated that the first messenger is outside of the cell, we're going to know that we're having a second messenger being involved in the inside of the cell, all right? So on the inside right here. So this is the first messenger. So let's say this is some sort of ligand, right? Some sort of peptide hormone. All right, so it's going to bind here, and what usually is inside the cell is that we have a G-stimulatory protein. All right, so this is going to be called, it's one of the many types of G proteins. All right, so it's going to be a G-stimulatory protein. So let me go and write that down in, I guess, green. All right, so we have a G-stimulatory protein right here, all right, that's already bound to this part of the receptor, all right, intracellular, so it's on the inside of the cell. Now, what happens the second we have some, let's say a peptide hormone, right, some sort of ligand that binds as a first messenger onto our G protein coupled receptor, what happens on the inside is pretty cool, right? So we have a series of reactions, or a series of steps, what I should say. So G protein, G coupled, the G stimulatory protein, right, on the G coupled receptor, G protein coupled receptor is actually bound onto GDP, all right? So it's bound onto GDP. Okay, so let me go and write that neater because I can't really read that. So it's bound onto GDP, our guanine diphosphate, all right? So that's the molecule it's bound to. The second we see that this hormone binds, this peptide hormone or ligand binds to this, our GDP is actually converted to GTP, all right? So we add a phosphate to this GDP because it only has two phosphates, hence the diphosphate, right? So we add a phosphate and bam, all right, we convert it into GTP and everything starts going awesome from here. So now we have GTP bound. Once we have GTP bound, our cell will have, or that G-stimulatory protein right inside the cell will have enough energy to travel along the membrane. All right, so our G-stimulatory protein will then travel along the membrane and it's going to try to find an effector enzyme. And so that effector enzyme is going to be located right about, like near it, all right? So let's say this effector enzyme, is drawn in red, right, is right here. All right, so this effector enzyme, say it's right here, and this effector enzyme is going to be called our adenylate cyclase enzyme, all right? So this is going to be our, just going to label it right here, adenylate cyclase, all right? So this is our effector enzyme. So it's nearby our G-stimulatory protein. So effector enzyme on the membrane, on the phospholipid bilayer membrane. All right, and so what this is going to do, right, once the G-stimulatory protein binds onto the adenylate cyclase effector enzyme, so we're going to see it bound onto here now, right? Our G-stimulatory protein traveled along the membrane and is now bound here with the GTP on it, right, with the GTP on it. And now what we're going to see is that the GDP or the GTP is going to get hydrolyzed back into GDP and a phosphate group. All right, so we're going to see that it dissociates again into a GDP and a phosphate group, right? So that the GDP now initially had the G-stimulatory protein inactive. All right, so let's go and write that down. Remember in the beginning we said it's initially bound to GDP, right? And so GDP, so let me go and write this down for you guys. So GDP keeps the G-stimulatory protein inactive, all right? But GTP, as you guys probably guessed, right? Guanine triphosphate keeps the G-stimulatory protein, right, active, all right? So it keeps the G-stimulatory protein, right, active. And instead of saying keeps, we can say it turns it active. All right, so it turns the G-stimulatory protein active once GTP is bound. All right, so it turns the G-stimulatory protein active. So right here, once the G-stimulatory protein is bound to the adenylate cyclase, GDP and phosphate and an inorganic phosphate is broken off from the GDP, and so that GDP goes back and it turns the G-stimulatory back to inactive. All right, so this in turn causes the G-stimulatory protein to be inactive, right, as it initially was, because now it's done its job. It has now binded to adenylate cyclase. The second that happens, guys, adenylate cyclase is an enzyme that does something really cool. It takes ATP, adenosine triphosphate, right, our main energy source in our body, especially in our muscles. So let me go and write this down. So adenylate cyclase like goes beast mode, right? So he takes ATP inside the cell. All right, so let me go and drag this over here. So this is adenylate cyclase doing this, okay? It takes ATP and converts ATP into cyclic AMP. All right. Now, cyclic AMP will then activate a protein kinase A inside the cell. So cyclic AMP will go ahead and activate a protein kinase A inside the cell. And now this protein kinase A will phosphorylate proteins inside the cell to carry out whatever cellular function it has to carry out. All right, so and we'll see that in like when we talk about what glucagon does, right, the opposite hormone of insulin, that what it does the opposite job of insulin, and we'll see how it acts on the fat cells and the glycogen in the liver, right, how it acts on the liver. It acts through this pathway, right, or even let's see, even adrenocorticotropic hormone, right, when we'll talk about the zona glomerulosa and the adrenal cortex. Right? So it acts in this process right here. All right, the protein kinase A is the main thing that we try to produce via a G protein coupled receptor. So this is the G protein coupled receptor, second messenger, because it's the so the G stimulated protein is a second messenger because it's inside the cell. All right, but this is different than the first messenger, is the ligand that binds outside the cell onto the G protein coupled receptor. All right, so this is G stimulated protein, second messenger, right? Intracellular mechanism to make our protein kinase A pathway, so protein kinase A can go phosphorylate, right? It can go and start phosphorylating. Its job is just to go phosphorylate proteins inside the cell to carry out whatever function that cell needs to carry out. All right, so phosphorylate, and it'll phosphorylate different types of proteins. All right, so that's the protein kinase A, second messenger, G-stimulatory protein, intracellular pathway. All right, that I wanted to get through in this video. So I hope that video made sense to you guys. Hopefully I'll see you guys in my future videos where I'll talk about more cool topics about biochem, physics, and chem. Make sure to like and subscribe and hit that notification bell. See you guys later. See you guys later, and stay safe.